Welcome to Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today, I'm talking with Amanda Schneeweis from Team Awatramani about their online data explorer tool, Dopabase. Hi, Amanda. Thanks so much for joining me today. Can you briefly tell us about your ASAP project and the impact that you hope to have on PD?
Absolutely. So first, I really like to express my thanks for the invitation to come on Discover ASAP. And just generally, Dopabase is fully funded by ASAP and wouldn't be here without it. So I want to acknowledge that first and foremost. So Team Awatramani's ASAP project is titled Redefining PD Pathophysiological Mechanisms in the Context of Heterogeneous Substantia nigra Neuron Subtypes. And so, let me elaborate on that. Our group has found that midbrain dopaminergic neurons are not homogeneous. In fact, they can be kind of grouped in these distinct subtypes based on their molecular makeup. And from work funded by ASAP, we've revealed that these molecular distinct dopamine subtypes appear to have different spatial patterns in the brain. They show anatomical and functional differences. I like to shout out that one subtype whose activity is correlated to locomotion exhibits selective vulnerability in Parkinson's disease. That's really exciting. But kind of like the impact that we hope to have on Parkinson's is that providing a detailed understanding of diverse dopamine neurons that exist in the substantia nigra in terms of their kind of like molecular signatures, their unique anatomical circuits, functional contributions to like motor behavior. We can really elucidate how the circuitry is actually disrupted in Parkinson's disease. So that's kind of the impact we hope to have.
Yeah, and I think this is super important too when you think about how heterogeneous the Parkinson's disease can be. That perhaps some of that could be due to this heterogeneity at the level of dopamine neurons as well.
Exactly. Yes.
Okay, so I mentioned at the top of this interview that we're going to talk about this data explorer tool, Dopabase, that you have created. So can you talk to me about how this tool is related to your ASAP project?
Yes, definitely. And so I really like to say that Dopabase is kind of like an outcome of the project, honestly. It really integrates a lot of findings from our like whole collective team into one tool, which is really great. And I know we're going to kind of dive into kind of the overview of what the tool does later. But so what I will say like in this moment is that the tool is great for like looking up gene expression kind of in like a moment's notice. Like it's been great. I've been having discussions, with lab members or my PI. We're in lab meetings and we're reading a paper. Let's look up that gene, and you no longer have to start up RStudio and kind of like code and create a plot and export it, which can just be a longer process. It's also been just this really great resource for lab members who aren't proficient in like computer science or coding. They have all these other amazing skills, but that's not one of them, and they can still query our data set easily. It's been really important for kind of like including all levels of trainees kind of into the conversation of RNA sequencing, and so that's been great. Kind of bringing everyone together into our ASAP project.
Yeah, it really, I think, is takes on the ethos of open science and making all of that data really truly accessible, regardless of your sort of level or ability to code or something like that.
Right.
Yeah. So I think you've led us really nicely into my next question. So I would like you to give us sort of a high level overview of what you can do with Dopabase, and include here also the kinds of data sets that people can take a look at in Dopabase.
Yeah, so Dopabase is it's an R Shiny app, and it uses the Shiny Cell package in R for those interested. It really enables users to, at the heart of it, it's querying midbrain dopaminergic neurons from wild type mice, and then we also have alert to G2019S mice. midbrain dopaminergic neurons. And so users can go into, it's literally dopabase.org, query gene expression or co-expression if they're interested in multiple genes, extract the information and generate kind of customizable figures, really without requiring any computer science expertise. Additionally, kind of excitingly, we just added some spatial information. So that is allowing users to identify the spatial location of the cell bodies of dopamine subtypes in the SNC. So it's really great. And as we've kind of said before, it's great for like all levels of users to go in and query that information.
Yeah, this sounds like such a useful tool for hypothesis generation, I feel like. And so, I'd love to hear more from you about, why you created this tool, if you had other sort of, use cases or advantages in mind, but beyond sort of just creating this sort of platform for hypothesis generation surrounding PD and heterogeneity of dopamine neurons.
Absolutely. So our lab totally aligned with ASAP on this. We're just really passionate about open science. And so the reality of developing a successful pipeline for sequencing, especially midbrain dopamine neurons, it's non-trivial, it's costly, and it's time consuming. And so we really realized that, many labs could benefit from this data set being available, and there's just no need to reinvent the wheel or expend resources on something that we did. And however, we really didn't want this data set to just simply be available. We wanted it to be really accessible to everyone. And so it was really important to create a tool with someone with like really minimal computer science expertise could use in query. And so, there are some really great tools out there that exist already. I want to acknowledge that. Absolutely. Our tool really focuses just on dopamine neurons, which are kind of notoriously more vulnerable to damage during the kind of like RNA-seq preparation. And so what we're hoping for is that Dopabase allows this figure generation for groups to use in kind of like grant applications, presentations, and that kind of like, as you were just alluding to, like there are these use cases that anyone who like needs like a plot generated can do that now with Dopabase. And really just the main highlight is that it's just supposed to be a data set that's really accessible to everyone.
Great. I love it. I think this distinction between making data available and accessible is a really key one. So I think that's great. So I would love to hear, we sort of have been touching on this point, but I would love to hear you talk more explicitly about what you think or what you hope that this Dopabase tool can contribute or will contribute to the Parkinson's field.
Sure. Yes. No, absolutely. So by providing this accessible way for labs to explore our sequencing data, Dopabase is really aiming to kind of enhance research efforts aimed at really addressing key questions in the field of dopaminergic neuron biology, kind of, and this really includes that selective vulnerability in PD, distinctive circuit functions of dopamine neuron subtypes. And then there's kind of this whole other, topic that I want to acknowledge is that there are so many labs that study PD from a completely different scientific angle than our team does in our lab. And it was really important to us that these teams are able to query this midbrain dopaminergic data set for genes that they're interested in that just might be completely off our radar. And this tool really ensures that any person, any lab looking into the genetic profile of dopaminergic midbrain neurons and how it relates to their own research question can do so and, move PD research forward kind of as fast as possible by getting everyone involved with, their own unique questions. And so that was really important to us.
Yeah, I think that, one of the reasons that we at ASAP here are so passionate about open science and sharing data is that, it's hard to anticipate the kinds of research questions or the use of a data set in the future. And so making it available so that other people can query it and sort of based on their own perspectives and interests is kind of vital for really accelerating sort of the discovery in the field. So I completely agree. Okay, great. So at this point, I like to end the interview by giving you the opportunity to share about anything else that I haven't asked you about today.
Sure. So yeah, just a little like plug for a Dopabase, dopabase.org. We're continuously adding to Dopabase. Our lab has some unpublished quantitative projection maps of these dopamine subtypes from the SNC to the striatum, and we're really working to get that uploaded very soon. That's been really exciting. We're hoping to add like a toolbox section for users who can browse kind of new intersectional genetic tools that are kind of continuously being developed by our team. And then lastly, like as our lab just collects more sequencing data, we're really committed to uploading that onto Dopabase for the PD research community to use. So yeah, really excited to, this is just the start for us.
Great. Thank you so much for joining me today, Amanda.
Thanks so much.